Hi, and welcome to this series exploring landmark experiments in protein science based on my book of the same name, which can be purchased on Amazon, and the link is in the description. In the last video, I talked about the discovery of proteins and enzymes, which are mostly classified by their physical properties such as solubility. They are known to have the general formula C40, H62, N10, O12, though some also had trace amounts of sulfur or phosphorus. A special type of protein called enzymes were found to be synthesized by organisms like yeast and enable the conversion of one type of molecule into another, such as occurs during the fermentation of sugars into alcohol. In this video, we'll continue on with proteins, but turn our attention to the discovery of protein subunits and the elucidation of the structure of proteins. In 1810, the English chemist William Hyde Wollaston was studying the composition of human urine and isolated an organic compound, that is, a compound that had carbon in it, that would later be called cysteine. However, the presence of small amounts of sulfur made it unusual, so it was suggested that cysteine was actually a double molecule connected by sulfur. He showed that this was correct when he treated the molecule with hydrochloric acid and found that it split into two molecules with a similar composition as cysteine but with very different properties. He called this new molecule cysteine, and yes, conveniently for everyone, they're both pronounced the same way. Almost a century later, in 1899, cysteine was isolated as a breakdown product of proteins. In 1819, Joseph Louis Proust wanted to find the molecule which gave cheese its unique flavor, so he took sheep's milk and skimmed it, and after draining it, he placed it in a jar for a year and a half. The milk curds, which he described as having a detestable odor, were then washed and dried until they turned into a syrupy solution, which had an excessively intense cheesy taste. Purification with alcohol resulted in white crystals, which he called oxyde casseur, and which contained ammonia, oil, and lots of carbon, similar to animal substances. Around the same time, the French chemist Henri Braconnot wondered if proteins, or albumins as he knew them at the time, would be converted to sugars when treated with acids the way starches were. So Braconnot boiled meat with dilute sulfuric acid and ended up isolating a substance which he named leucine, from the Greek white, due to its shiny white appearance. But it turns out that this was the same substance that Proust had previously isolated, and in fact, Braconnot himself isolated Proust's oxyde casseur in 1827 without realizing it was the same as his leucine, but had renamed it aposipedzine. But in 1839, Mulder repeated these experiments and realized that aposipedzine, oxyde casseur, and leucine were all the same molecule, and it was the name leucine that stuck. The same year he isolated leucine, Braconnot also discovered another molecule when he boiled gelatin in dilute sulfuric acid and named it sugar of gelatin, since he found it to be sweet to the taste. It was renamed glycine in 1858 due to its chemical similarity to leucine. By the late 1800s, several more of this type of molecule had been isolated, all containing both a basic amino group and an acidic carboxylate group, and so this family of molecules was appropriately called amino acid. There are 20 amino acids that make up proteins, and the part of the molecule that isn't either the amino or the carboxylate group is called the side chain, and this is the part that makes each amino acid unique and gives them their character. It was quickly surmised that amino acids were the building blocks of proteins, since the hydrolysis of proteins, which is a process where molecules are broken down with water, resulted in the formation of amino acids, but no one had actually proven this by synthesizing proteins from amino acids. The first real evidence that amino acids were indeed the building blocks of proteins emerged in the late 1800s, when Theodore Curtius observed that glycine ethyl ester formed a biamino acid ring structure called glycine anhydride. However, it was Emil Fischer who subsequently figured out the mechanism of protein synthesis. Fischer noticed that Curtius's glycine anhydride was chemically just one step away from being a molecule with two amino acids if only the ring structure could be broken and stabilized. So, he treated the molecule with ethanol, which produced the glycyl glycine peptide he wanted, but he also found that the reaction went through an intermediate molecule that readily condensed back to glycine anhydride, so the reaction was not very efficient. Ingeniously, Fischer realized that if he stabilized the molecule with a glycine derivative bookmarked by two chlorine atoms, called chloroacetyl chloride, and then added ammonia, then the N-terminal glycine in the peptide would react with a glycine derivative, which would displace one chlorine atom in the process, whereas the ammonia would then react with the N-terminal of the intermediate molecule, and this would displace the other chlorine atom, and all this would result in the formation of a stable 3-amino acid peptide composed of 3 glycine amino acids, or diglycyl glycine ester. And this insight allowed him to synthesize for the first time long chains of amino acids, which he termed polypeptide, and whose characteristics could not be distinguished from natural proteins. Until the early 1900s, proteins were generally thought to be colloids, that is, small molecules which randomly aggregate into large complexes, and the Sweden-born Theodor Svedberg was convinced that this branch of chemistry would ultimately help to explain the processes of living matter, but that a lack of a reliable method for the determination of molecular weights of substances possessing a very complicated structure has been a serious obstacle in the progress of our knowledge of the chemistry of the proteins. And so in 1912, Svedberg realized that the usual method of sedimentation, which was basically just to let things settle at the bottom of a tube, would benefit from the stronger centrifugal forces offered by a centrifuge, but the instruments generated so much heat that the resulting convection forces pushed the particles down the sides of the tube faster than the forces generated by the centrifugal force. So Svedberg and his student Hermann Rinde redesigned the centrifuge to address this and other issues. To help control the heat generated inside the centrifuge, they replaced the air around the rotor with a constant flow of hydrogen gas to decrease friction. They isolated the rotor from the bearing using a screen of copper in addition to circulating water around the bearings surrounding the rotor shaft. And finally, they sprayed oil on the tooth wheel assembly. To ensure that the instrument spun at a constant speed, which is necessary if you want to precisely measure sedimentation rates, they added a special clutch to efficiently translate the rotational energy from the toothed wheel, which is attached to the motor, to the rotor. There was also a window that was mounted on top of the cell, which is where the sample was loaded, and which was illuminated by a light below the cell via a prism so that the samples could be monitored in a separate room above the centrifuge through an opening in the ceiling. In addition, a camera was mounted above the cell window to more precisely measure the average size and distribution of particles. With this instrument, Svedberg could begin to determine the molecular weight of proteins using hemoglobin, which is a protein in red blood cells that binds iron, as his first sample. The molecular weight of this protein was suspected to be at least 16,700, though this varied wildly depending on the source, from a fraction thereof to multiples. And at the time, Svedberg was still convinced that proteins were colloidal substances, so he expected hemoglobin to separate into multiple bands of low molecular weight when spun down in a centrifuge. But to his surprise, his experiments consistently indicated that the molecular weight of hemoglobin was 4 times 16,700. Further experiments confirmed these results, and analysis of other proteins revealed that some even registered in the millions. Therefore, these experiments showed with little doubt that proteins were large, stable molecules and not colloids, and refuted a paradigm that existed for nearly a century. As we saw earlier in this video, by the 1900s, Fischer had shown that amino acids were linked together via peptide bonds, but many remained skeptical as late as the 1950s. An important contribution to the resolution of this controversy, and to that of the structure of DNA, as we'll see in the next video, was the discovery of X-ray diffraction in 1912, when it was found that X-rays could be diffracted by a crystal in a pattern based on the molecular structure of that crystal. William Thomas Asprey explains, "The object of using X-rays instead of, or rather in addition to, visible light is, of course, to extend the range of vision, i.e. to permit objects which are far too small to be seen by the human eye, even with the aid of the most powerful microscope, to be viewed at least mentally. Just as ripples are seriously distorted by pebbles which would be far too small to disturb the form of ocean waves, so X-rays, some 10,000 times shorter in wavelength than visible light, are seriously distorted by atoms and molecules which are far too small to disturb the main form of waves of visible light." Things are seen through the waves which are thrown back from the body looked at, and only such irregularities can be distinguished as are not much smaller than the wavelength of the waves used, because irregularities which are negligible compared to the waves make negligible impressions on them. The most carefully machined and polished surface is quite rough from the molecular point of view, even though by ordinary light it may appear so perfect as to be optically flat, but by using smaller and smaller wavelengths, smaller and smaller irregularities can be detected until finally, in the light of the X-rays, after making the appropriate calculations, the shapes of the atoms and molecules themselves can be distinguished. And so in the 1920s and 30s, Asbury and his colleagues used X-ray diffraction to study over 100 X-ray photographs from a wide range of material under as great a variety of conditions, including human hair and sheep's wool, which are made up of the protein keratin, and concluded that they were almost certainly built up in a rather imperfect manner of molecular chains running roughly parallel to the fiber axis. In addition, he found two separate molecular structures for these proteins, a natural unstretched form, which he called alpha keratin, and a stretched form, beta keratin, which he could extend up to 3 times its length by exposure to water or steam. Asbury also found that hair has the striking property of always returning to its original length if wetted after extension. However, in a number of images, much of the center is obscured by a halo of high spacing, which clearly arises from the scales of the hair. So they developed a method for stripping the cuticles and scales from hair by using the strands as the strings of a wire bow of the cello pattern and drawing it rapidly backwards and forwards through coarsely powdered glass under a slight pressure. When they imaged simple molecules like cellulose and silk, they found that the crystalline aggregate formed by the fiber molecules are long and thin and lie with its long axis roughly parallel to the fiber axis, and that the molecules are straight, fully extended line patterns. Not surprisingly, they found that the more complex molecules, such as cotton or wool fibers, produced much blurrier images and required a higher degree of crystal purification, but the general parallel nature of the molecules could still be seen. They also noticed the difference in patterns produced by unstretched and stretched wool molecules, which indicated that, like hair, the fiber was present in 2 separate forms. From these images, Asbury calculated that the distance between the intramolecular pattern in alpha-keratin was similar to the distance between the hexagonal glucose residues in cellulose, whereas the distance in amino acids in beta-keratin was slightly shorter than the fully extended pattern of silk, which indicated to him that unstretched wool must be built of the same chains as stretched wool in a folded state, i.e., the mechanism of its unusual elasticity is simply that of a molecular spring. He also found that the sulfur atoms and cysteine residues of unstretched wool and hair interacted together, which are now called disulfide bridges, and that basic and acidic residues were connected, which we now call salt bridges. Importantly, these experiments provided additional support for Fisher's polypeptide hypothesis, that is, that proteins are a sequence of amino acids linked by peptide bonds, since the images clearly indicated that the amino acids were connected to one another. As mentioned earlier, the purity of the crystal determined how well the images could be analyzed, and Asbury's crystals were not yet good enough to analyze proteins of high complexity. However, John Desmond Bernal and Dorothy Crowfoot Hodgkin proposed that a knowledge of the crystal structure of the amino acids is essential for the interpretation of the X-ray photographs of animal materials, and in studying the structure of amino acids, they were able to produce the first clear images of proteins, describing pepsin as dense globular bodies, perhaps joined together by valency bridges, but in any event separated by relatively large spaces which contain water. Which meant that we are left now with a paradox that the pepsin molecule is both globular and also a real or potential polypeptide chain system, and the immediate question is whether the chains are formed by metamorphosis and linking up of the globular molecules, or whether the initial unit is the chain itself, which is afterwards folded in some neat manner which is merely an elaboration of the intramolecular folding that has been observed in the keratin transformation. In 1936, Linus Pauling, who was considered by many the greatest chemist of the 20th century, suggested the theory of protein denaturing, like the change in appearance of egg white after it's been cooked. He attributed the different properties of individual proteins to their uniquely defined configurations, and therefore, denatured proteins are characterized by the absence of a uniquely defined configuration. And so he proposed that proteins were denatured when exposed to excess heat, alkali, acids, or alcohol, which are conditions that were known to lead to the loss of protein function, coagulation, and changes in their chemical properties. He also concluded that one of the major chemical forces holding proteins together was the hydrogen bond, which I talked about in the first video on chemistry, where a hydrogen atom acts as the glue between two electronegative atoms like oxygen and nitrogen. So it was proposed that one of the first consequences of exposure to denaturing agents was a disruption of the weak hydrogen bonds, which results in the random association of amino acids side chain to each other, and which in turn meant that the protein lost its unique configuration and consequently its specific properties. In 1937, Pauling set out to decipher the structure of alpha-keratin with a plan to use my knowledge of structural chemistry to predict the dimensions and other properties of a polypeptide chain, and then to examine possible conformations of the chain. However, he eventually reached the conclusion that a piece of the puzzle was still missing and stopped working on the problem for 15 years. When he started working on this again, he first started by assuming that all amino acids are structurally equivalent in terms of how they could be folded in a polypeptide chain, and inspired by a theorem of mathematics, which states that the conversion from one asymmetrical object into an equivalent asymmetric object could be accomplished by one rotation about an axis followed by a translation along that axis. He set out to determine if this type of transformation could result in hydrogen bonding between amino acid residues and keratin. Fortunately, the number of ways that this transformation could occur was limited by the fact that the alpha carbon of an amino acid is in resonance with oxygen and nitrogen, and so this peptide bond had to be planar. And so to help visualize the process, Pauling sketched the amino acid chain of keratin on a piece of paper and tried to fold it in a manner in which hydrogen bonds formed between the residues. He only found 5 possible configurations, but only one of these agreed with the known data, and that was that of the helix, which he suggested was a major component of many proteins. He also proposed that amino acids could form lateral hydrogen bonds with residues from a separate chain running in parallel, either in the opposite direction forming a beta sheet, or in the same direction, which he called pleated sheet. In this configuration, the hydrogen bonds between the two chains are perpendicular to the plane of the sheet, directing their carbonyl groups in one direction and their amino groups in the opposite direction, and all the chains are oriented in the same way. And so to summarize, by the early 1900s, most amino acids had been discovered, and Emil Fischer had shown that proteins were made up of a string of amino acids linked up by peptide bonds. Svedberg used centrifugation to show that proteins were large molecules and not just colloids. Asbury used X-ray diffraction to study the structure of proteins and found that they could extend like a spring and that amino acid side chains interact by salt and disulfide bridges. Bernal and Hodgkin found that proteins could be globular and wondered whether they were synthesized like this or folded after the amino acid side chain was synthesized. And finally, Pauling suggested that the properties of proteins were determined by their unique three-dimensional configuration, primarily due to hydrogen bonds between amino acid side chains, and the loss of this configuration results in the loss of their function. And so this brings this video to an end. I hope you enjoyed it. And if you did enjoy it and would like to know more about this topic, please check out my book, Landmark Experiments in Protein Science, using the link in the description. And if you have any questions, please leave them in the comments and I'll do my best to answer them. And I hope you'll join me for the next video on the discovery of DNA, its subunits, and the elucidation of its structure.